Alright, so what we're going to be doing in this lab is we're going to be looking at the effects of antibiotics, or inversely, antibiotic resistance of bacteria. So what I'll be doing is I have my M8 plates here, and also my cotton swabs, sterile cotton swabs, that I'll be using to spread S. epidermis or E. coli onto my plates. Once I have my lawn spread out, so inoculating my plate and spreading, what I'm going to do is I'm going to take my antibiotic discs and place them onto my inoculated LB plate. So in your manual, it has you normally do 3 per plate. What I'm going to be doing here is 6 per plate. Just so we can see, and then in our results we'll be looking at the zone of inhibition, which is kind of analogous to antibiotic resistance. So the smaller the zone would be, the more resistant the bacteria is to that particular antibiotic. So to get started, what I'm going to do is open up my sterile vet and my epidermis, and I'm just going to simply put the cotton swab in here just to soak the swab. And then what I'll do is take my bacterium, just spread this across. And the reason I'm rotating the plate as I am is because I want to have a full lawn of growth. So rotating the plate helps ensure that occurs. So another way that you could do this is if you had liquid broth and you transferred about 100 microliters or 0.1 mils, depending on where your bacteria growth is at, and you could use a spreader. So I'm going to close this up now, and this is now waste, so you want to put it into a proper waste container. And now I'm going to take my other sterile swab, and inoculate my LB auger with my E. coli. Again, rotating my plate as needed. That way I can see that I've spread across the entire plate. And one trick that I use is I use the reflection of light off of the plate to see if there's anywhere that I've missed. So, one thing that's going to be really important and useful for doing this is you want to use some forceps. So these guys will make it a lot easier when placing your antibiotic discs. So I'm going to just start by adding all my antibiotics onto my E. coli plate. Okay? So again, working in my sterile environment, I'm going to just simply pick up a disc, and then place it onto the plate here. So one thing I normally do is I give it a little tap to make sure that this disc will stick to the auger. So remember, when we grow bacteria, we invert our plates, so you want to make sure that your disc does not fall off. So that's why I give it a gentle little tap to push it down into the auger. So now that I have all of these on my E. coli, so what I'm going to do is now plate all the antibiotics on my S. epidermis plate as well. So this won't be shown in the video since it's going to be the same process as what we did. So one thing that you want to make sure to do, and I may have not done the best job at this, is making sure to spread your discs as far apart from each other as possible. Since you'll be doing this on 2 plates, this will be a lot easier to achieve in comparison of 6 discs on 1 plate, as I have it.